This lecture will get into some of the introductory concepts related to genetics and some of the basic biological processes that happen that allow genetics to occur. So this unit is going to be largely about the idea of heredity, which is the transmission of traits from one generation to the next. And in most reproducing organisms, variation is key to this process. And we're going to learn about how this variation is created. Variations are differences in the appearance between offspring and their parents and their siblings. And genetics, broadly, is the scientific study of heredity and how variation occurs. So, in a literal sense, children don't really inherit physical traits from their parents. It's actually information to provide those traits in the form of genes that are inherited. And those genes are passed on to the next generation from parents to offspring via specialized reproductive cells called gametes, which are your sperm and your eggs. And genes are units of heredity that are made up of segments of DNA. And as we've learned, your DNA is packaged into chromosomes, and any one length of chromosome can contain 100 over 1,000 different genes all along its length. And the specific location where you might find a gene along a chromosome is called its locus, or loci, for the plural. So humans have 46 chromosomes, and that's made up of 23 pairs. And these are found within what we call the somatic cells, which are all the cells in the body except for the gametes, the sex cells. And here we see what's known as a karyotype, which is a ordered display of all those chromosomes showing their pair. So this certainly isn't how you find the chromosomes within an actual cell. This is just a way of organizing them and seeing what the chromosomes are and what their pairs are. So you receive a chromosome from each parent, and each one of those chromosome pairs is called a homologous pair. And homologous, as we'll get in the next slide here, basically means very, very, very much similar, but not exactly the same. And so they're similar, they carry the same genes that control the same inherited characteristics, but there might be some variations in those, the coding of those genes that give you the variety. What we see here is actually a pair of chromosomes that have gone through the process of DNA replication. And so the replicated chromosomes are connected to each other in these pictures by these structures called centromeres. And these duplicates of each other are called sister chromatids. Right? So typically in a non-dividing cell, you'll just see one strand of these, and in fact they won't even be in this form. This is their condensed form when they're going to be going through the process of being split up by the cell either during mitosis or meiosis. So just a kind of an analogy of what we mean by homologous. So here's two different homologous pairs. In this case, here's a gene for making pies, but here's an apple version of the pie. Here's the cherry version of the pie. Here's another version of a gene that gives the instruction for making cookies. Here's an M&M version of cookie. Here's a chocolate chip. So this person has two different versions of these genes for making cookies and pies, but they have the same version of the gene for making these cupcakes. See there. And then here's another homologous pair, and it has its own set of genes. What you might notice here is both of these versions of these genes both make the same identical looking jelly beans, but in this case, this version of the gene makes more of it. So in the next unit, we're going to talk about gene expression and what controls gene expression. So we'll get into this sort of stuff then. Now, of those 23 pairs, there is one pair that's special, and these are the sex chromosomes that determine gender. and these are called the X and the Y chromosomes. So human females have 2 X chromosomes, and human males have an X and a Y chromosome. And the other 22 pairs of chromosomes are called autosomes. Okay, so again, each of those pairs of chromosomes includes 1 chromosome from each of your parents. And so you've got 2 sets of 23. 1 set came from your mother, 1 came from your father. And so a cell that has 2 sets of chromosomes, 1 from the mother, 1 from the father, is called a diploid cell. And we use the symbol 2N to denote a cell that has such a number of chromosomes. So for the case of humans, our diploid number is 46, or our 2N number is 46. Now, in a cell in which DNA synthesis has occurred, each replicated chromosome consists of 2 identical sister chromatids, and I showed you on that earlier slide. So you'll see them in this format where you'll have 2 sister chromatids which are identical to each other, connected together by those centromeres. Now, haploid cells are the sex cells, the gametes. They have just a single set of chromosomes, and we call that haploid, when you just have 1 of each chromosome, and that is given the symbol N. So the haploid number for humans is 23. And so it makes sense, if you're going to make a diploid cell, the mother and the father's gametes are going to combine together, each with their 23, to reconstitute a diploid cell, which has 46. And so the process by which we go about creating those haploid cells from the diploid cells of the body is through the process of meiosis. And we'll get more into details of how that works in the upcoming slides here. So there's specialized cells within the reproductive organs called germ cells that go through this process. So basically the cycle of life for sexually reproducing organisms is spelled out here. So you have haploid cells with just 1 of each chromosome fused together during the process of fertilization, creating a 2N zygote, which then reproduces that cell through mitosis, the many millions and trillions of time to create the full adult organism. And then some of those select cells then undergo meiosis to create the next generation of gametes, which can then go on to form fertilization for the next generation of organisms. Okay. So, and that's basically what's spelled out here too as well. Okay, so that cell that results from fertilization is called a zygote. And basically, if something goes wrong with this process, particularly with the formation of the gametes and the proper splitting up of the chromosomes, then you may not end up with a proper number of chromosomes during fertilization. You might end up with 1 too many or lacking 1. All right, so the process of meiosis is all about trying to reduce a cell that has a diploid number to a haploid number. And so there's a lot of parallels between mitosis and meiosis, except that meiosis has 2 rounds of cell division called meiosis 1 and meiosis 2. And at the end of meiosis, you end up with 4 daughter cells instead of just the 2 that you see in mitosis. And again, another parallel or contrast, I should say, to mitosis is that at the end of meiosis, you end up with cells that have just half as many chromosomes, just 1 of each chromosome. Okay, so just a real quick summary of the steps of meiosis here is, again, you've got pairs of homologous chromosomes. So here we have 1 for dad, 1 for mom. They undergo a round of duplication of the DNA. And then it goes through 1 round of meiosis where you then divide up those homologous chromosomes. And then you go through another round of division of the chromosomes where you then split up them to just having 1 of each chromosome. So in a way, it almost seems like we're going backwards, right? We want to take, this cell that has 2 and end up with cells that have just 1, but we actually start off by doubling the number. And then we have to go through these 2 rounds to reduce it back down to 1. But the end result is we end up with 4 cells that have just 1. And there's some other things that happen, though, that make this advantageous to go through this seemingly extra step. And a lot of that leads to the variety that helps sexually reproducing organisms compete and evolve. Okay, so here is the steps of meiosis. And again, you're going to see a lot of similarities between this and mitosis. So you have an interphase where the cell's basically hanging out doing whatever it should be doing. Usually if this is a germ cell, it's in some arrested state within the sexual organs until it receives a signal that it needs to go through meiosis. Okay, and so once that signal is received, the cell will duplicate the DNA, and so now you have 2 copies of each chromosome held together by these centromeres. And then the cell enters prophase 1, and the chromosomes will condense. Okay, and so instead of being long and stringy pieces of DNA, they condense into these more compact forms, and the nuclear membrane dissolves away. And then something that happens in meiosis but not mitosis is those homologous chromosomes will sidle up next to each other and form these structures called tetrads. And then something very special happens. Again, this is unique to meiosis. Is during that formation of those tetrads, you get something known as synapsis, where pieces of the neighboring chromosomes on these homologs will cross over and swap pieces. So this is called crossing over with synapsis. And so now you can see at this particular length, this chromosome has a chunk of chromosome and variations of genes from this chromosome here and vice versa. So if this was a chromosome from dad and this was a chromosome from your mom, you've now gone and created these hybrid chromosomes that are a combination of genes from your dad and your mom all in one. Okay, so then this happens with all the chromosome pairs, all the homologous chromosome pairs. Again, in this model I'm showing you, I'm just showing you 2 homologous pairs. It would be really tedious to show this and too busy with all 43 or 23 pairs, I should say. All right, so the next thing is the centrioles move to opposite poles of the cell and create these spindle fibers. That's part of the spindle apparatus. And that helps align the chromosomes along the equator, this imaginary middle line down the middle of the cell, moving up and down in this case. And then, so that's metaphase, when they meet up in the middle. And the next stage is anaphase, where the homologous chromosome tetrad pairs are pulled apart. Okay, so you end up with, again, kind of what you start out with here, and the fact that you have, 2 of each chromosome separated apart. And then you go through a telophase where you create another membrane around each of those separated groups of chromosomes. And then there's a cytokinesis where you then split into 2 cells. Okay, so essentially we went from a 2N cell to a 4N cell back to a 2N cell here. But you can see we still have 2 of each chromosome, although in some cases they might be a mixture of genes from the homologous pairs. So then we go through into another prophase in meiosis 2. So again, just like prophase, the nuclear membrane dissolves away, and then we go into a metaphase where the chromosomes will line up and a new spindle apparatus will form. And so then the spindle fibers, again, will then help to pull apart the homologous chromosomes, the sister chromatids apart in this case. So this is very much similar to what you see in mitosis. Okay, so this is the stage of anaphase where you pull those apart. Okay, and then we then go through another telophase. It's telophase 2, where we make a nuclear membrane, nuclear envelope around each of those groups. And then we wrap up with cytokinesis, where we split each of those 2 cells now into 4. And so now the end result here is we just got 1 of each of these chromosomes, whereas we started with 2 of each at the whole beginning of the process. Now, there's a little added twist here. So what I just showed you explicitly is what happens in males and the production of sperm. And that's the process of spermatogenesis, is what that is called. In females, though, the germ cell that ultimately goes through meiosis, called an oocyte, doesn't divide equally at each of those cell divisions. And so you end up at the end getting these 3 smaller haploid cells that don't have much cytoplasm at all to them, and 1 large cell that has most of the cytoplasm that was within the original cell that develops into the egg cell. And so basically what this does is it creates an egg cell that has a lot of the nutrients and all the organelles that it's going to need to quickly, once it gets fertilized by a sperm cell, to start dividing and going through mitosis and all that. By the way, the scale of this is way off. Sperm cells are much, much smaller than an egg cell. They would be like a little period next to the egg cell in terms of size. Okay, so in a comparison, mitosis, meiosis. So mitosis, the goal end of mitosis is to create cells that are pretty much identical from the parent cells. They're going to be genetically identical. They're going to have the same number of chromosomes as the parent. However, meiosis, the end goal is to create haploid cells, just 1 of each chromosome, as opposed to the parent cell had 2 of each that was diploid. And we also end up with cells that are genetically different from each other than from that parent cell. Okay, so some other differences between mitosis and meiosis is, again, mitosis only has 1 round of cell division, only 1 round of dividing up the chromosomes. And basically, meiosis 2 is what's most similar to mitosis. As you can see, in meiosis 1, that first round of splitting up the chromosomes, the homologous chromosomes line up side by side and get pulled apart. Whereas in mitosis, all the chromosomes line up end to end, and the sister chromatids are pulled apart. Okay, that's pulling apart of the sister chromatids doesn't happen in meiosis until the second round, until meiosis 2, when you pull these apart and then create the haploid cells. And then another obvious difference is the crossing over event that happens during meiosis. Okay, so all this is part of how we get variety in sexually reproducing organisms. So first of all, we get different types of genes, different versions of genes, what we call alleles, through mutations. So mostly due to mistakes made during the replication of DNA, do you create new alleles of genes. So that's the original source of genetic diversity. And then we reshuffle and mix up those alleles during this process of meiosis to produce the genetic variation that we see in the final organisms. So there's basically 3 main mechanisms that contribute to this genetic variation during meiosis. So it's the independent assortment of chromosomes, crossing over, and random fertilization. So independent assortment has to do with the fact that when those homologous pairs of chromosomes basically line up next to each other during prophase and then metaphase in meiosis 1, it's more or less random as to which side of the equator they line up on. And it's independent. So for each pair of chromosomes, it's independent of all the other pairs. And so in the case of humans, since we have 23 pairs, that makes 2 to the 23rd possible combinations of chromosomes lining up during metaphase. So that's more than 8 million different ways that you can arrange those 23 pairs. And so basically what this looks like is, so you've got those, here we're looking at just 2 homologous pairs here. So one possibility is you could have, say, the blue ones are from the dad and the red ones are from your mom. So you could have, these 2 of the dad lining up on the left and these 2 of the mom lining up on the right. But another probability is to have this combination, where you've got one from the dad here, one from the mom here, but the other homologous pair, you've got the one from the mom on this side and the one from the dad on this side. And so when they go through metaphase 2 and then at the end of the production of the daughter cells, you can see that in this case here, you've got this mixed up combination of chromosomes that originally came from the dad and the mom within the final gametes here. And this is again just for 2 homologous pairs that we're looking at here. When you got 23 of these and all kinds of random possibilities of them lining up either on the left or the right and getting pulled towards the left or the right and separating out, that's where you get that 8 million possible combinations. And then throwing into this is the whole crossing over that we covered earlier. So there's an average 1 to 3 crossover events happens per chromosome. So that is going to further mix up that combination of genes that were originally from your mom and your dad into the chromosomes of your gametes. Okay. And then, so you've got, all these, over 8 million possible chromosome combinations just from independent assortment. And then you got all that crossing over that happens. And then it's pretty much random as to which oocyte and which sperm are going to fuse during fertilization. That calculates out to about 70 trillion different possible diploid combinations of zygotes that could be created. And even more when you throw in the crossing over that can happen. So, this is how we get that unique variation that happens in each zygote. And even within a family where, you're sharing, half of your genes between you and your siblings, there's still a large amount of variation that happens because of that randomness of which gametes come together. All right, so that's it for that topic. If you have any additional questions, please ask in class.